very happy to be here to present Ziccum. We are the modern alchemists, indeed, because we are helping to unlock the biopharma revolution. We are enabling new drug delivery forms. We are solving every drug stability issue. And this is really unlocking everything that can be done with biopharmas. We are enabling groundbreaking innovation. I think you all know that in pharmaceutical development, the most focus and investment these days, it's all about biopharmaceuticals. That's the big topic ahead. And there's been a real revolution now because there's a complete new era in pharma when the RNA field is opening up. Of course, you have all heard of mRNA that became possible due to the enormous need in the COVID pandemic. This is a genetic coding for drug remedy, and it's opening up completely new possibilities across indications. But the platform comes with very severe limitations. For the pandemic, everything was accepted, right? Because the need was so tremendous. So governments and states and industry put in anything they could to make a vaccine possible. But now to use this fantastic platform for new drugs, for new indications, it really needs help addressing delivery and addressing stability. And this is what we do. The Ziccum technology is all about enabling new drug delivery forms and solve stability issues. We do this for biologics in general and for mRNA specifically. And we have now extensive, validated, and fantastic data generated for biologics in general, for mRNA more specifically. Before we go into that, just one word about the company. We are a spin-off originally from Karolinska Institute in Stockholm. We are very happy to have the inventor, Professor Per Gärde, still on the board. He's still an associate professor at Karolinska. We have a really high caliber team in the company. I have been recruiting international co-workers. So we have a multinational, diverse team supporting Ziccum. We have our financial manager, Christo Vassilev, here today. Also very happy to take questions from you. My background is very significant. I spent 30 years now in management positions, mainly building really large corporations in the US and in Germany in pharmaceutical development and pharmaceutical manufacturing. Now back in Sweden, I have been managing several clinical stage biotech companies, but Ziccum is even more exciting, I'm happy to say. Because we have this huge need in industry of delivery and stability, like mentioned. And I think this is the key slide. It's a very quick presentation today, right? But let's take a good look at this one. What are we doing in Ziccum? We are turning liquid biologics into thermostable dry powder. That sounds really simple, right? You have a simple picture here, going from these super fragile COVID shots into a robust, thermostable, particle engineered perfect powder. But the benefits, there's so much value here to industry. Speaking to industry, this is obvious more or less, but I think it's really good for investors to understand all the value steps here. So the base is to avoid the cold chain, the cryogenic chain. Instead of having this super expensive -80 treatment that the COVID vaccines needed, we create a product that can be stored in room temperature. So there's enormous cost gain, there's an enormous benefit for sustainability. Vaccine campaigns have normally an enormous carbon footprint, and we take away 99% of that carbon footprint. There's a lot of cost benefit if you could find an alternative method for drying. We are still have better yield and lower expenses in our process. Then we have a very different benefit, because once you have created a really well-designed powder drug, then you can give new administration forms. Then you can give it by inhalation. You can even consider tableting into oral drugs. So this opens up for completely new treatments with new drug forms. As a total, we lower the cost so much that you can suddenly address new indications. So unmet patient needs where there are no treatments today can now be met when we lower the cost a lot for these fancy, exclusive mRNA molecules. And this has been the existing Ziccum focus. Now we have one additional value here. I will present very, very shortly today, because in our work to optimize the performance of our technology, we have come up with very surprising data that we can also increase the performance of drugs. So we'll get back to that very quickly. A quick view, just what is the old version, what is the old model? The old school pharmaceutical drying technology is freeze drying, lyophilization. That's rarely applicable for biologics. It's difficult, it's low tech, you have no process control, you have no way to control the way your particles look when they come out. As an industry, pharma has moved to spray drying. This works very nicely for small chemical active ingredients, but it ruins any biologic. There are so many stress factors, mechanical stress in every step, and there's a lot of heat stress. So it's important to understand that laminar paste, our technology, is completely different. It's not a tweak of this. And in industry, people have a hard time to even grasp this because it's so different. It's a unique technology. We had the innovation done a number of years ago, and we have optimized it since then. And it's now working, and it is working for any modality. So very quickly, what is this technology? It's a drying equipment. We have countercurrent flow, so we have optimized flow rates and temperatures. So this is really high-class chemical engineering, you can say. And I love it because I'm a chemical engineer originally. And you have really efficient drying because it's based on mass transfer. It's not heat transfer. So we are able to take away the solvent without any heating, without any freezing. And that is unique. It's done at room temperature. And it gives a product which is perfectly dry, very much meeting industry requirements. We have done this for all kinds of modalities, like mentioned. Proteins, peptides, enzymes, monoclonal antibodies, and a number of vaccine platforms. Recently, we have focused a lot on mRNA, because this is the top of the value pyramid. It's the most delicate entity, and it's also the highest value entity. So every gram, every molecule, every dose we save here, preserve, is giving a lot of value to the pharmaceutical client. The base application is that we perform this drying, you get the product in dry form, but then the client would reconstitute, put it back into liquid for injection. So that's a base case. On top of that, we have excellent data for inhalation. And this is really a value sticking out of the ordinary. Nobody has achieved inhalable mRNA in LNP so far, globally in the industry. I attend a lot of conferences, and in Frankfurt in July, for example, there was an mRNA conference discussing that it's impossible to inhale LNP particles. And I had to say, well, hello, we have done it. We do it. We have perfect properties for this. So these are the two key routes. And then in the future, you could also consider oral dose, like I mentioned. Speaking of milestones, we have a lot of milestones. I focus on only two slides here. One is that we have animal data. So apart from our partner studies, we have performed a number of studies to give proof, good proof for industry. We have done animal studies where we have perfect preservation of the mRNA activity in mice. The CRO we are working with, they were quite stunned by such extremely good results. And then we get to this special topic. It's only one single slide here because time is so short. We have far more information to give if you're interested. So, like I mentioned, we have now, by accident, you could almost say, found out that our technology is not only preserving the mRNA efficiency and activity, but it's even increasing it a lot. And we have achieved this for several different formulations. The lipids, that's the fatty acids who build the LNP particle around mRNA. And we have now made repeated trials. We have proven this in great detail. We have found the structure, morphology it's called, the structure of the LNP changes when it's treated with laminar paste. And this enables a better release of the mRNA in the cell. This is now tested in vitro, in live cells, where we measure the protein expression. So the next stage now obviously will also be animal trials on this topic. And like I mentioned, we have a lot more information to share if you're interested. Then quickly, we have these technical milestones. I think we don't have time to review them again. On the market potential, we have an ambitious plan now. We have our own master plan to develop the equipment to final stage, optimize the performance. We have a 3D simulation project running for 3 years. When I joined Ziccum, I could see this is completely new technology. How can we develop this? It will take forever if we just stay in wet trials. So we now have digital modeling by an expert team in Zurich in Switzerland, modeling everything in computer cloud environment. So using AI to mimic the performance of the system. So we can now generate a lot of data digitally as well as in wet trials. And carefully developing the organization. Practically, the rig is a plug-and-play unit, so to be easily implemented in existing industry facilities. The business model is licensing. Right now we are performing paid studies for world-leading partners, aiming, of course, at licensing agreements with partnering, co-development, and then the licensing with upfront payments, milestones, and then royalty. We have signed a lot of agreements here, 5 agreements signed in the last year. The last one is with Recode Therapeutics, which we're able to name disclose. We have good revenue created. We have a pipeline of dialogues. We have signed with top 10 big pharma, world-leading mRNA COVID biotech corporations. And we have a number of other platform addressed as well. I think it's good to know we are not just focusing on mRNA. We are also working on protein pharmaceuticals and on viral vector vaccines. And finally, the addressable market. This is a very conservative estimate. It's just the Western markets. Looking at the mRNA development, looking at the vaccines we address, and then saying we would only get 1% to 2% royalty. So it's a very conservative estimate. And then our addressable market is $600 to $1,800 million per year. So that's the market we can capture. That's the market we are targeting. And that's it.
Thank you so much.
Thank you.
Okay, so first question. How will the increased mRNA activity after laminar's pace processing, I'm sorry, you have to listen, there is a long question. How will the increased mRNA activity after laminar pace processing,
yeah.
Thank you, impact Ziccum's market positioning?
I mean, if I can speak freely.
Do it, please, always.
The company value has multipled. It's multiple now with this possibility. Because mRNA, I mean, normal pharmaceuticals, you have to produce a chemical or a biological molecule, right? And you have to put it into a formulation, and then you have to see how to administer to patients. With mRNA, you're just given mRNA coding for the treatment. And it's the patient's own body that produces the treatment, right? So it is possible to treat any kind of disease with it. So that's the starting point. However, to get the mRNA into the body, it has to be formulated in LNPs, lipid nanoparticles, or similar nanoparticle structures. And they have a small limitation on how much mRNA you can put in. But if we increase the release of mRNA, instead of just using 5 or 20% of this precious, expensive content, you could instead use maybe 90 or 100% of the content after our treatment. That opens up for completely new medications.
Okay, I'll end with a critical question then.
Yeah.
Did you not previously aim to get vaccines to the third world countries, and is that still the overall goal?
That's a really good question, I think, because when the company started out, there was very little data, but there was this unique mass transfer drying, right? And the initial business model was to enable vaccines for global distribution. That was before the COVID pandemic. That was before mRNA took over the world, so to say. And that is still a very good purpose, but we can reach high value much quicker if we address mRNA. So we do both, but we go for the best money first. Yeah.
Okay, that will conclude our session. Thank you so much.
Thank you so much. Thank you.